and drugs and rock and roll and chips and clips and chains and whips. Peggy Brown, that was my name. Watch me now, I'm going down, down. 'Cause I'm crawling and falling every morning, rocking and rolling, keep holding my head and down.
Ladies and gentlemen, your host for this evening, Quentin Cooper.
Hello, good evening, welcome. Yes, I am Quentin Cooper, tweeting as @QWERTY for these purposes, which I'm very proud of as a tweet name. So welcome along to the Famelab UK Final 2016. You probably knew you were here. Can I first of all just check how many Game of Thrones fans we've got in tonight? Yeah, that's very noble of you. And how many Spurs fans? None. Okay, that's good. So basically, the Game of Thrones fans were prepared to sacrifice the chance to watch the first episode of the new series. The Spurs fans are off watching the TV now. Okay, that's good to know. And also, anyone here a Famelab virgin? Not just an ordinary virgin, I'm sure there are plenty, but anybody absolutely new to Famelab? Okay, welcome. Thank you for coming. I should probably explain. So this is something that goes back to 2005 at the Cheltenham Science Festival. We had this idea of getting people to talk about science. Participants were given 3 minutes to enthuse us, not confuse us, and possibly amuse us about some aspect of sciences and scientific thinking. And it proved surprisingly, amazingly popular. Helped by the British Council, it spread like a kind of benign virus, infecting ever more countries round the world. So now in 2016, which, by the way, for binary fans is 11111100000. 6 ones, 5 zeros. Thought you'd like to know that. It's also a triangle number, the first since 1953, when Elizabeth was crowned, so also worth thinking about as well. Anyway, so since now in 2016, 1111110, anyway, yeah. It's now the world's biggest science communication competition. It's in dozens of countries. CERN has its own version. NASA has had its own version. And now you're here tonight for the UK element of this world's biggest science competition. And I should say, it's on every continent apart from Antarctica, and we are working on Antarctica quite hard. Yesterday, I was in Madrid for the Madrid Masterclass for the Spain finalists. And all of the winners will get to go to Cheltenham in a few weeks' time, in about 6 weeks' time, by which time 3 Starks, 2 Lannisters, and a dragon will be dead, to take part in the international finals. 27 countries taking part. That is more than UEFA Euro 2016, which slightly unfortunately clashes with the Cheltenham Science Festival. We'll deal with that later. And tonight we'll decide who gets to represent us in the finals. And by us, I mean, of course, the entire United Kingdom, in the old-fashioned sense of the word, not affected by any recent referenda. Now, each of our 10 finalists has already proven their skills at serving up irresistible bits of bite-sized science. They've all battled their way through regional heats and finals, winning out over tough opposition and a limited range of backstage snacks to be here tonight. But only one of them will walk away as our champion for 2016. And as a small added incentive, they will also get £1,000 in cash. Woo! Thank you very much. £750 to spend on public engagement activities and their body weight in courgettes. We also have 2 runners-up, joint runners-up, who each get £500, semi-woo, £300 of which is to spend on a public engagement activity, excluding paintballing and crown green bowling. And there is one more prize which you, lady there in the front row, third along, yes, you two, you get to decide on. Okay, along with the rest of the audience, okay? So that is the audience prize. I'll give details of how you vote later. But before I give those details and before I introduce our judges who get to decide on the rest of the prizes, it's only fitting that since this whole shebang started at Cheltenham, we introduce the supreme being of Cheltenham. Well, their technical title is CEO of Cheltenham Festivals. Please welcome Louise Emerson.
There's nothing to make you feel nervous than by being called a supreme being. But anyway, thank you all for coming tonight. Welcome to the Famelab UK Final. And as any of you who have been to Famelab before will know, the audience is a really important part of Famelab. It's all about performing in front of a live audience. And I do believe, although I can't see all of you, this is a live audience.
Mostly.
Proof. Like Quentin just said, all of the contestants tonight have been competing over the last few months in the regional heats, and I would really like to thank all the people who work really hard to put on the regional heats. So thank you very much for that. And then the winner, of course, will come to the Cheltenham Science Festival. So date for your diaries, 8:30 on the 9th of June. I'm not sure if that clashes with a football match or anything like that, but of course it's going to be much better than a football match. So put it in your diaries, come and support the winner tonight, and of course do much more, because there's lots of stuff going on at Cheltenham Science Festival. It's going to be, as usual, a really good show. It finishes on June the 12th. And the only other thing...
That's not the Famelab final. It's not that long. It's the whole festival, right?
It does start the day before, though. So the semi-finals are the day before. It just remains for me to thank our sponsors, the official sponsor, EDF, and our partner, Hazeltine Lake. It's really important to have sponsors for this. They really help us make this happen. And the only other thing, then, is to tell you all to enjoy yourself tonight. It's always a good night. Have fun.
Louise, thank you. And as is appropriate for somebody whose empire also includes a literature festival, Louise is bookending this evening, as well as appearing at the beginning. She'll be back later when we come to the grand prize. But who, of course, will win that grand prize is not down to her. It's down to our three judges. So let's get them up here. First, he's primarily a primatologist, but his work's also evolved into evolutionary biology, and he's presently presenting various TV series, including BBC 4's Secrets of Bones. It's Ben Garrett. Next, we've someone who takes Neil Young's magnum opus, Rust Never Sleeps, very seriously. She's an expert on corrosion, working on the nanoscale to help make materials more durable, and also finds time for side projects like helping to restore the world's only surviving Dornier 17 bomber after its remains were found in the English Channel. Please welcome Imperial College's Professor of Materials Science and Nanotechnology, Mary Ryan. And our chair is writer, actor, TV science presenter, former Bangos the theorist and fan of Werner Herzog, often found down in sewers or at the top of high towers, and recently returned from Kazakhstan, where he covered Tim Peake's launch live for the BBC. It is a rare appearance in London at ground level for Dallas Campbell. And he's wearing the actual suit in which he was buried there as well. Now, Ben, the finalists get 3 minutes each. They can make points powerfully, but they can't use PowerPoint. They're only allowed any props they can bring on by themselves. So mostly, what this is going to be down to is what they say, plus how they deal with their questions. So what are the official criteria you'll be judging them on?
The official criteria we're looking at tonight is... Being good. Being good, yes. A big thing is charisma. Well, obviously this is Famelab and very science heavy, but we want charisma in there. We also want clarity and we want content.
Clarity, content, charisma, fine. Now, Mary, all of the people, they've won through their heats, they've won through their regional finals, so it's fair to say they've got reasonable content, reasonable clarity, reasonable charisma. What sort of thing is going to make the difference between an also-ran and a winner tonight?
I think for me, I want to see enthusiasm, and I want to get the sense that they love science, because that's what I do.
It'd be kind of disappointing if they didn't love science.
But there's a difference between doing it in the lab and being able to communicate that kind of connection with an audience, how much you have a connection to your science, and that's a big challenge.
Great. And Dallas, the official criteria is that Famelab is open to anyone over 21 and working, studying in science, technology, engineering, medicine, or maths.
Yes.
I can't help but notice that
me.
you would not fit that criteria, technically, because you come from a non-sciencey background. Should Famelab be open to people even if they don't have a science background, as long as they're passionate about science, like we've just been saying?
Well, I'm under 21, obviously.
Obviously.
So on that criteria, then I might do it. Well, it's a good
So
question.
you've already got numeracy issues right there, yes.
Yes. No, you're right. I mean, I think going forward from Famelab, if going on to present science shows, which is what I do, it would be a terrible thing if it was only scientists who are allowed to talk about science. There is still a kind of this sort of hang-up that only scientists can talk about science. So no on that front. But Famelab is very specifically for scientists. So yes.
Good.
Or
I'm glad we clarified. Good. Having put okay, well they do have a tough thing to do tonight because they've got to really decide on one winner and 2 joint runners-up from some expert fields. So can we please give a round of applause to thank our judges in advance for their work? And later on, once the judges are away, speedily reaching their verdict, we could just let you wander the streets of Southwark, but instead we have a real treat. We're going to have our minds poisoned by strychnine queen Dr. Catherine Harkup, author of A is for Arsenic, The Poisons of Agatha Christie. That is still to come. Right, I've explained the rules. We've met our judges. We've mentioned the interval act. We've locked the doors. Time to bring on our first Famelab finalists. And just a reminder, all of these people are reasonably new at this. They've never performed in front of an audience as large and pungent as you before. So you can G them up and spur them on by the noise you make. So be loud. And remember, that will encourage the judges. The judges are all involved in the media, therefore they're insecure, therefore they can be swayed by you, the audience. That's the way how it works. Now, the sequence has been randomly determined by laser etching the names of all 10 finalists onto protein blocks, feeding them to lab rats and noting the order in which they consume, which, by the way, is exactly the same method they now use for the Eurovision Song Contest. So first, and very popular not only with rats, we have Alex Connor from the University of Birmingham Medical School. Never the Birmingham University Medical School, because that would make the acronym BUMS, and that just wouldn't do. Alex says he splits his time between hiding from his research team and trying to remember how all the bits of the body work 5 minutes before a lecture. And his fear is that someone will eventually find out and make him do a grown-up job instead. On Twitter, he describes himself as leader, entertainer, fantasist, jobbing higher educationalist, and science things. I'm not quite sure which science things, but we're hoping to find out after 3 heart-stopping, head-popping minutes with our first Famelab finalist, Alex Connor.
On December the 29th, 2013, the Formula One legend, Michael Schumacher, was skiing with his family in the French Alps when he fell and he hit his head on a rock. His brain swelled and he suffered neurological damage from which he has yet to recover. He joined an unenviable list of more than 60 million people every year who suffer some form of traumatic brain injury and brain swelling with devastating consequences. Welcome to Famelab, everybody! I hope you're looking forward to a fun evening. We study medical biology, which means we want to understand what is going on. But it also means we want to try and do something about it. To understand brain swelling, know that the heart is constantly pumping blood up to the brain, about 25 litres an hour. So the brain needs some kind of flood defences. And when you have a bang on the head or a stroke, those flood defences start to fail, and water comes gushing from the bloodstream into the brain, causing the brain to swell, and that causes damage. That's because the skull is very hard, it's a very fixed volume. A swelling brain is not. The brain has 100 billion neurons. To put that in context, 100 billion is how much it's going to cost you guys in London to buy a house. That isn't true, but it is how many stars there are in our entire galaxy. And that number is eclipsed ninefold by the number of cells of the brain that are not neurons, that regulate what comes in and what goes out. And that is very tightly controlled. If you've ever tried to get into Legs 11 in Birmingham wearing jeans and a t-shirt, I haven't. If you have, you'll know the strict control. the flood defense cells of the brain, shaped like stars, magically called astrocytes, and they are very small. I could fit 10,000 of them in a single file across the palm of my hand, and if this Greenwood Theatre was your brain, every astrocyte would still only be as big as a grain of salt, and it gets smaller still, because every grain of salt astrocyte has thousands of tiny gateway proteins that let water in, called aquaporins. And in our lab, we study the shape of a single one of those gateway proteins, and we block them. So when you have that bang on the head or that stroke, water can't come from the blood of every heartbeat into the astrocytes, and can't cause the brain to swell. And with those drugs, we're hoping to win the battle of hearts and minds. Thank you very much.
Alex, thank you very much. That was excellent.
Thank you.
That was terrific. You're obviously incredibly passionate about the brain. You talk about it in this wonderful way. What was it when you were, if you cast your mind back a few years, what was it about this particular area of biology that got you so excited?
I study proteins and the shape of them. And that's interesting, scientifically and research-wise, but what they do and how to fit keys into those shapes and fix something that affects us all, people we love, that it doesn't really matter that it's the brain. I'm happy to work on the heart and the kidney and some of the lower bits I forget the names of. But anything really where we can fix a problem, that's basic medical science.
And have you always been interested in this since you were a kid? Or is this a later thing in life?
It's a really good question. Yeah, I keep waiting for somebody to tell me to go and get a proper job as an accountant. So yeah, always.
Yeah, people are telling me to get a proper job too. What you've described is a lot like, the needle in a haystack, right? This is a tiny cell in a huge organ. How do you start to isolate these kind of cells and receptors and proteins? What tools do you need to be able to do that?
It's very difficult. What you need to do is work as part of a massive team. And luckily, science is like that, isn't it, with publishing? And what we do is we look in the literature and we saw that mice without these proteins, if you bang them on the head, I didn't do that. If you bang them on the head, they didn't get brain swelling. And we all went, oh, we think we can stop that protein without taking our protein out of a human. We think we can get in there and block it up with a drug that you eat. And that was the it wasn't like, there's an American called Verkman, I feel I should mention him because
That's okay.
far better than I'll ever be. But yeah, his idea, our reading of his idea.
Very quickly, as a scientist and a science communicator, your topic affects people. My mum had a brain hemorrhage, my granddad died of a thrombosis. Is it easier or more difficult to talk to an audience when something that you talk about is much more tangible? I mean, Dallas, his crazy stuff up in the sky, but no idea what he's on about half the time. But yours affects people in this audience. Is that harder for you or easier for you?
It's a risk, and you have to have empathy. Early on, I didn't have empathy occasionally, and you upset people, and you learn from that because you realise, hang on, this is people. And when you think like that, when you realise how would that feel, the second that happens, it's the easiest job in the world.
Okay, empathetically tell your neurons to bang your hands together. One more time, please, for Alex Connor.
Thank you.
And if you take one bit of science away from tonight, it's never trying to get into Legs 11 in Birmingham wearing jeans and a T-shirt. Now, second up, we've our reigning Scottish champion. Well, he won the Edinburgh Heat, so that's good enough for us. And it's Frank Machin, a PhD student in plant molecular biology, keen gardener and collector of carnivorous plants. Frank tweets as @necrobiology, mostly about plants, but there's the odd intriguing other one, like, I role play with my desk. It's as dirty as it can be, and I just have to sit there and watch. Oh, yeah. Leaving that image aside, if you can, Frank says he spends his days in the lab using lasers and other tools to probe how plants develop, and his nights moonlighting as a biotech entrepreneur trying to grow flowers in new ways. He gardens, he moonlights, but is he a late bloomer? Let's hope the stage is fertile ground for Frank Machin.
So how many of you guys remember The Hitchhiker's Guide to the Galaxy, the radio series? Yes, nerds. Well, in that show, there are a race of aliens that have a peculiar quirk. When a need arises, they evolve really quickly. So, for example, if they're reaching for some sugar to put in their coffee, as they go to reach for it, they'll evolve and become something that has giant arms. Oops. That has giant arms. And we all know the biology geeks in the room are probably screaming. I can hear you, I want to swear. The biology geeks are screaming, that's not evolution, not even a little bit. And they're right. That's transformation. That's changing the shape of your body. That's developmental biology. And what's interesting about them is it's actually pretty similar to the way plants grow. Now, animals, they have a fixed body plan. We get 2 legs, 2 arms, and a head, and then we get bigger. Plants are constantly developing. They're in a state of flux. A new leaf here, a new branch here, new roots down here. And at some point in their lives, they'll develop a flower, an organ they've never developed before. And this isn't just an odd thing plants do to keep me in a job. I wish. This is important to their survival because if an animal needs food, if I need food, I have to get up off the couch and I have to go to the fridge and I have to stand there like this for about 20 minutes. A plant is stuck where it is. So it has to respond to its environment by changing the way it develops. So if it needs food, it has to produce more leaves so it can photosynthesize for sugars, or it has to produce more roots so it can extract nutrients out of the soil. Now, this plastic development, as we call it, the ability to keep changing your shape, means that if you look at a plant, you can see its entire life history in its body plan. You can see where one branch was obviously growing into the dark and didn't do very well, so it stopped growing or has been damaged and fallen off. You can see where the roots have gone down into something solid because they've stopped and had to develop a different way. And this is totally different from anything animals do. Can you imagine what it would be like if you grew a new arm whenever you needed one? But back to Hitchhiker's Guide to the Galaxy. The aliens, when they reach for something, when they develop their giant arms, they might develop into something that can't drink the coffee. The arms might not be able to bend at the elbow. They might not be able to drink anything. And this is the same for plants. When they're stressed, they can develop into something that can't grow as fast. And this is a big problem for us, because if they can't grow as fast, we can't get as much food. So this is why we study plant development, to look at better ways to grow crops and to feed the world. Thank you very much.
Frank, thank you. Of course, it would be very nice if we could just grow a new arm when we needed one. Is there something else we can also learn from plants that can help us mimic that kind of behavior?
To mimic that kind of behavior?
Yeah, you know. Can you tell me something from plants that will help me grow a new arm if I lose this one?
Well, we really don't want to be able to mimic that behavior because a large part of who we are requires our body to stay the same way. Can you imagine? So your brain cells have to stay basically where they are in order to hold all your memories and everything. If they could redevelop at will, you would lose all sense of yourself. So plants being able to develop like this is really the only way they can survive. We are very lucky that we can move around, and if it's a particularly hot day, we can go inside to an air-conditioned room. Plants have to sit there and take it.
They do. So growing plants is not a fast process, generally. And I guess what you're trying to do is speed it up. But how do you go about designing your experiments to study those processes?
Well, the first thing you look at is how the plants grow normally. So typically everyone uses just one plant, and we use mouse ear cress. It grows along the pavements. You've almost definitely seen it, but never really thought about it. But we use that because it's small and simple and it has a very predictable way it grows. And essentially the way we look at it is we take a lot of photos. And if we want to go into more detail, look at the molecular stuff, we cut parts of it off and extract it. Because you can do that to plants, whereas the ethical problems come with animals. You also have the problem that mice typically, if you cut off their arm, they won't grow a new one, but plants will grow a new thing. And so you can study the way they grow that new stem or root, which means you can study the way they adapt to their environment and find out what's important for this, which helps when you're breeding crops to breed in new traits. And that's going to become more important as the climate changes, as environments become more hostile, really, to plants. We're going to need crops that are tougher and more resilient in order to survive this.
Thanks.
Very good talk, thank you.
Welcome.
You're saving the world through increasing agriculture and da-da-da-da.
Well, not me personally.
I'm marking you down for that. I thought it was. I actually thought you were. You're not.
I do my bit, but let's not pretend I'm saving the world.
Oh, right, okay.
Otherwise, we're one car accident away from damnation. Let's not all rest this on my shoulders. There are others, better people who do this.
Thank you for your honesty.
I'm not going to lie to these people. Paid good money to get in here.
Does your work, which is does have a global impact, does it trickle down to my granny who likes to grow flowers? Is this citizen science? Does this, the stuff that you're doing in your labs behind biohazard control, does it affect these guys with not the wheat they eat, but the flowers they grow on their doorsteps or things in their gardens?
The science I do is 90% sort of exploratory. We just want to know so we can know. 10% of it, I work with companies to help develop approaches to deal with this in the real world, to identify genes they can concentrate on breeding, to identify things they can use as biostimulants. These things, when they eventually get on the market, can usually be bought and sold in garden centers, and you can apply them to your flowers. But typically, you can spend more time caring for your flowers than a farmer who has, say, several million crops to look after, and your granny, who I assume has not nearly that kind of land.
No, I think she does.
She grows them really close together.
Well, that's probably a bad thing to do anyway. You really shouldn't grow things too close together.
I'll tell her.
Well, yes. But I have to say, this is very typical when you tell someone you're a plant scientist. First thing they ask, gardening advice.
Okay, I hate to break up this burgeoning Ben Frank double act that's developing here, but one more time, please, for the terrific, part Triffid, Frank Machin. That'll teach you to try and compliment somebody, Ben. Yeah, you won't be doing that again in a hurry.
It doesn't often happen anyway. It won't happen again tonight.
Our third person singular is ex-Brisbaneite and self-confessed vision nerd, Kate Storrs. Kate just been awarded her PhD and started as a postdoc researcher in the MRC Cognition and Brain Sciences Unit at Cambridge. Only with the science crowd, ladies and gentlemen. There's a whoop for the MRC Cognition and Brain Sciences Unit. Woo! Which she says means she can continue getting paid to think about why some things look like cats and others don't. There is a little bit more to Kate's work than dividing everything she sees into cat and non-cat. She wants to understand how the brain turns points of light on the eye to what we recognize as faces, flowers, phones, or indeed felines. When not being a visionary, Kate is a weekend artist, currently learning how to draw like a 15th century engraver. For now, though, she has just 3 minutes to try and etch herself into your memories, and with luck, your visual cortex. Lots of applause with your paws for Kate Storrs.
Consider the fable of the minnow and the tentacled snake, or how the animals got their cleverness. In the shallow river, brimming with snapping, biting, minnow-eating things, sadly had to live the minnow. Despairing at all that was brimming, the minnow grew 2 nerve cells in his head, one on either side. Of course, you and I call these Mauthner cells, but the minnow's not to know that. Each one crosses over and has a tail as long as the minnow's that ties to all the muscles down that side. These 2 nerve cells make the minnow ever so nervous. Every time he feels a ripple or a tickle, or hears a wriggle, or catches a glimpse of spittle on his right-hand side, his right-hand cell sends a zing of electricity down all the muscles on his left, and he coils up like the letter C and darts away. And so his Mauthner cells keep him safe from all the snapping, biting, minnow-eating things in the shallow river. Until one ill-fated morning he meets the tentacled snake. The tentacled snake is the laziest snake in all the shallow river. She lies bent in a crook from dawn till dinner time, like a folded-up umbrella. moving not a ripple, but using her tentacles to feel for anybody else's ripples. By and by, the minnow ambles along and finds himself in the crook of her neck. A peaceful place, it seems, but with one tentacle she's felt him ambling by. Aha! She twitches her tail, he coils up and goes to dart away, but instead he darts straight into her mouth. Because, although the minnow is clever, the tentacled snake is cleverer. The moral of this story is that the way animals got their intelligence was through an arms race. It took 3 billion years from the first single-celled life to the first fish-like creature that had an escape reflex. 2 cells wired together to make a little electrical circuit. And then it took only perhaps another 200 million years for something like a tentacled snake to evolve to exploit the reliability, the predictability of that fish's reflex. The snake is also hardwired, its hunting behavior, but that requires a vibration sensing circuit and a tail twitching circuit and a mouth opening circuit, perhaps thousands of nerve cells, all wired together in just the right way. Now, consider us. The human brain contains about 90 billion nerve cells. You can make a lot of electrical circuits out of that. They give us the ability to make choices that are so free and so flexible that even we can't predict which path we'll take. Clever us. In the words of the philosopher Daniel Dennett, yes, we have a soul, but it's made of lots of tiny robots.
Thank you, Kate. Very good. Could you not make a tentacle snake model?
I considered. It was hard enough to find a carp on eBay.
As you mentioned us towards the end, is there anything in us that's as hardwired as your little minnow there, that if X happens, Y responds, without us having any control? That fundamentally...
Absolutely. We have spine-mediated reflexes, so if you go to the doctor and they tap you on your knee and you sort of involuntarily kick, the thing that makes you kick, the nerve signal travels from your knee to your spine, just in your back, and then back out to your knee muscle. There's no involvement of your brain at all, so if you equate yourself in any way with your brain, there are certainly things your body does that don't involve your brain at all.
Did you enjoy doing that?
I did. It was great fun. I read all of Rudyard Kipling's Justice.
I was going to say, I read Daniel Dennett. Tell me about Daniel Dennett. I just read Consciousness Explained. So I've got sort of Dennett in my... Well, I was interested, are you a fan of Dennett? He's sort of quite controversial, I think. And I was just wondering where you stand with Dan Dennett.
He was a large part of why I went into vision science. I first did an arts degree, and I'd always been interested in the mind and consciousness, but I think I always felt that it wasn't legitimately in the purview of science, hard science. And then I spent a sort of gap year reading a lot of Daniel Dennett and Francis Crick and Douglas Hofstadter, and they persuaded me that you really can scientifically think about consciousness and free will and so on. So I'm very grateful to him. I don't necessarily agree with everything he says.
Is it Dennett as a writer, as a storyteller, as someone who conjures images?
I think he has a nice sort of manifesto for how we can do science about things like consciousness. He says, maybe there's some sort of mystical hard problem that we can't approach yet, but we know there are also lots of little problems, like, when I flash a light that's very, very dim, and sometimes you see it and sometimes you don't. What's different in your brain? What's going on differently in your visual system when you see it from when you don't? And maybe studying really little cases like that, which is the sort of thing I do, can give us an inroad into this sort of bigger, more mysterious thing called awareness or consciousness.
Okay, just a quick one. You mentioned,
quick.
I'll be very quick. Choice. Is choice really a good thing? And do you think we really can't predict what we're going to do, or do you just think we don't know enough about the brain to understand how to predict what we will choose?
Well, possibly we live in a deterministic universe, so if you had enough information. But
really quickly.
Yeah, sorry.
Yeah.
I know. Sorry.
It's a simple one, really.
I won't
Sorry,
mention the
simple
inverse
one.
then.
Simple one.
Also, notice this is a judge who's got to decide on a winner, asking if there's such a thing as choice. Which is slightly worrying as well.
Yes, absolutely.
Yes, there is, and you can choose her if you want. Okay, a wriggle, a spittle, a ripple of applause, please, for Kate Stores. And I should explain for anybody who wondered about that squeaky, wheezy noise in the front row you heard. That is if they reach exactly 3 minutes. It's not somebody with some sort of asthmatic condition. Right. Now, fourthly, we have the winner of our Hull Regional Final, Manish Kuruvilla. He's a PhD neuroscience student at St Andrews in Scotland and originally from India. So how Manish ended up in Hull might seem curious. But since his particular interest is spatial memory and navigation, he can doubtless explain. When not finding his way around our heads, Manish says he has a peculiar interest in the creative storylines of world wrestling entertainment, and in adding to his memory vlog, which has short videos spanning everything from the latest neuroscience to interviews with Canadian rock musicians and Scottish comedians. Given his navigational expertise, it should be straightforward for him to orient himself through the next 3 minutes without losing any of us along the way. Prepare to follow Manish Kuruvilla.
If I asked you to leave the room right now and come back in a few hours, how would you locate the chair you were sitting at? In fact, how did you make your way to the Greenwood Theatre tonight? Or better yet, how do you manage to not get lost every single day? 2 words that aren't Google Maps. Place cells. Place cells are cells in your brain that tell you where you are. If we placed electrodes into the brain of a rat, specifically the hippocampus, and recorded cell activity as the rat explored a maze, we'd see certain cells being active when the rat was in certain places, to the point where these Nobel Prize-winning cells would have created a mental map for the entire maze. So tonight's question is this: how do place cells go from being active to creating this mental map of the world? Like this. They use arm movements, whether we turn left or right, and combine that with the landmarks we see around us. Now, we've all been there. It's day 1 of that vaguely exotic holiday. You're trying to get from your hotel to a restaurant, and the receptionist hands you a map that typically bears no resemblance to the real world whatsoever. You start your journey, and place cells begin to be active as you turn left or right. But at the same time, they're anchoring themselves to the landmarks you see along your way. So if you went back to the restaurant the next day, you'd think of that journey not just in terms of left and right, but as left at the Starbucks and right at the post office, and so on. Now, if you're still a bit lost, think about this. If you regularly use GPS, you'll be familiar with the feeling of going to a place, maybe for the 3rd time, but still feeling unsure of the route you need to take. And here's why. GPS biases us towards focusing not on landmarks, but purely on turning left and right, which disrupts the ability of place cells to create these mental maps, which in turn affects our ability to find our way. So the next time you use GPS, pay attention to the landmarks you encounter, especially at decision points like roundabouts, and see how that improves your navigation. I'd like to finish with that timeless navigational hymn, Amazing Place. Amazing place, oh, Nobel crowned, that saved a wretch like me. I once was lost, but now I'm found, thanks to its spatial sensitivity. 'Twas place cells that taught my brain to learn the places I'd already been. How precious did place cells appear. The sat-nav failed me. Thank you.
Manish, thank you very much. That was terrific. Did you have a moment in your life where you were going to go left for music or right for science? I wondered. I could, because you're accomplished at both.
Thank you so much. I actually did. The summer before I joined university as an undergrad, I did a summer course at Berklee College of Music in Boston. I was very fortunate to do that, and I realized how terrible I was compared to the 12-year-olds. And I think my life choice was pretty made after that. So, thank you.
So I did leave this room earlier, and I went about 15 meters in that direction. And then to get back here, I think I traveled 250 meters around the building. So the question is, I think I'm the only one allowed to ask this, do women really have a bad sense of direction? Am I just atypical?
I feel like there are two parts to the answer for this. For the first part is for the sake
Be very careful.
Yeah, exactly. The first part is for the sake of how tonight goes. I'm going to say no. In fact, women are better.
It could just be me.
No, we don't really know whether these gender differences are accurate. They tend to be stereotypes very often. This idea that we develop these cognitive maps is across genders, and it's irrespective.
Good talk. I'm still weirded out by the guitar. In a good way. It's all right. Is there something, as you briefly touched on it, that we can do to improve our ability to orientate ourselves? Does that affect us at a cellular level? Is it just learned behaviour that we improve how we are, or can we fundamentally actually develop what we are?
So from a neuroscience perspective, I'll take you through the little journey, because I'm a bit of a history nerd when it comes to these things. In the 1940s, a behavioral scientist came up with this ridiculous theory that we have this map in our brain, purely on behavioral data. And then over the last 50 years, the neuroscience of this has kind of developed the evidence. So you have these place cells that fire when you're in specific places. You have head direction cells that orient the kind of direction you're facing like a compass. You have grid cells that act as like a grid almost to track how far you've traveled in your environment. Some cells pay attention to big landmarks versus small landmarks. So the brain seems to be well programmed to pay attention to these from a spatial perspective. And if you think about just beyond navigation, right? If I was writing the speech, trying to remember it, it's all a bit of navigation. Whether you're writing something, you're navigating through not just a physical place, but something in your head as well. So it makes sense for the brain to possess
like
these
memory palaces, people talk about.
Absolutely. So the memory palace is a fantastic example of that. And yeah.
Sorry, Quentin. Quentin's hovering.
Can we please thank Manish Kuruvilla for something spatial? By the way, oddly, he temporarily lost direction there, which is and Mary, I can draw your attention, it's still available as a download, is the Radio 4 series called Men Read Maps and Women Gossip, co-presented by myself and Viv Parry, which looked at myths about male-female differences.
I know, I have.
And they found they were all nonsense, except the orientation one.
Oh, but there's no also
reading
throwing.
maps, yeah.
Throwing, apparently. Girls really can't.
We didn't do it. Maybe Series 2 when we
at
come
throwing.
back for that one as well. Finalist 5 is Robert Thompson, a research fellow based in the London Centre for Nanotechnology and the Electrical Engineering Department of UCL. That would be the same London Centre for Nanotechnology where Mary is on the academic staff, so we're going to watch out for special interest
watch out,
here.
but I am Imperial and he is UCL, as I understand.
It's
so.
still the same thing.
So it might be worse.
We're just making you declare an interest. It's important we're squeaky clean here as well.
Can I just say I don't work?
At all. We know that about you, Dan. It's absolutely true. You're just lucky.
clear that up.
Now, we're not studying the fundamental properties of materials by shooting laser beams about, as he puts it. Robert can often be found in a pub being fun and friendly with a beard. Presumably his beard, it's not clear. As well as being fun and friendly, he's also frequently frank, singing Sinatra covers and show tunes in a bar in the West End and or playing his saxophone. He says Famelab is a great opportunity to combine performance and science, something he doesn't get to do very often. Tonight, though, no classics, no cover versions, all original material from Robert Thompson.
I've got with me in this bag some pretty old tech. Yet it's still one of the best ways of harnessing the sun's energy. And by pretty old, I mean over 3 billion years old. Yes, it's a plant. Plants can convert the sunlight they absorb into energy for photosynthesis with 90% efficiency. And they do this while being flexible and biodegradable. something that material scientists are endeavouring for in new electronic materials. Now, plants are able to do that because of a little-known particle called an exciton. You see, when special molecules in the plant's leaves absorb the energy from the light of the sun, that energy is taken on by an electron in the molecule. And this electron, given more energy, jumps up from the other electrons, leaving behind a hole. These electrons are negative. Therefore, relatively, this hole here is positive. And our electron, negative, is attracted to that hole. Let's use an analogy. You, sir, could you please just stand up here for me? Excellent. So this can be our electron. And with a little bit of energy or encouragement, I've got him up out of the seat that he's left behind there, and we'll call that the hole. So, sir, if you just turn around and look at the audience, you'd like to get back down into that seat, wouldn't you, and out of the limelight? So there's some attraction there between our electron and the hole. And it's this attraction that creates the exciton. The hole and the electron are one particle, indifferent. They are an exciton. Wherever the electron or the hole move through the material, because of the attraction, they move together. Thank you, sir. Now, in the plant, this exciton, when it moves about as one single particle, it takes with it the energy that it's absorbed. This energy can therefore be transported around the plant and can be used for creating sugars or converting carbon dioxide into oxygen. But the exciton can also be used in new materials made of plant-like molecules for electronics. And we call these materials organic electronic materials. Organic electronic materials use the exciton, and the exciton, once created, can then be used to create electricity in things like solar cells. And this process can happen in reverse, where the exciton created by electricity could then go and emit light later in things like TVs. Now, these plant-like molecules for organic electronics can be made into inks and plastics, and this creates the opportunity for a whole new technological landscape. I want you to imagine solar cells that are cheap and can be rolled out over the rooftops of South African wilderness. Or I want you to imagine TV screens that can be printed like newspapers. And it's that reason that because of the exciton, the plant is leading a technological revolution.
Very good. Thank you, Robert. So I've been warned not to ask you too many technical questions about nanotechnology, because that's not fair. But one thing that is interesting is that plants aren't really that good. They're not very efficient on a molecular level, but there's an awful lot of energy, human energy, to replicate what plants do. Can we even get as good as plants, which aren't very good?
So, as far as I'm not a botanist or a plant biologist, so I don't know the inner workings of plants. And me and Frank had a little chat before about what I was saying, if it was true or not. It's definitely true what I'm talking about. But it's true that the whole process of photosynthesis, once you get past that original exciton exchange level, is a bit lossy as it gets later on because of the enzymes that are used and losses there. However, that initial process, when you can get from the original absorption to transferring the exciton, is actually pretty good.
For one photon, one wavelength.
Yes, for one
huge
photon
solar
for one
spectrum
wavelength.
that's wasted.
There's a lot of the spectrum that isn't used, but we can now, using different types of material, tuning this exciton as to what it absorbs, can make that far more efficient.
Okay.
Can we get as good as plants? Well, plants have a lot of hidden secrets. One thing that's recently been discovered is that actually plants use quantum physics for some of this transfer as well. And there's an effect called quantum correlation, which means that the excitons in there can no longer be considered as separate things. They actually know what each other's doing. And therefore, the energy transfer in a plant is not as random as one might imagine. Now, if we can continue to learn about plants and take ideas like that, then I imagine one day in the future, yes, we can get something that is close, if not at the efficiency of something like that.
Thank you.
Thanks for that, Robert. I didn't write anything down because I kept thinking, don't write anything because you lose concentration. I don't understand what you're talking about. I work with bones. I'm so easily pleased in life. But what you work with and what you study is obviously very applicable and very relevant to us in everyday life. do you find it hard, again, similar to a question earlier I asked, do you find it difficult to get from quite complex science? I found that quite difficult, very well explained, but do you find the transition to get from that complex science to, and there it's on your TV? Do you find it easy to explain that? Because I nearly wrote something and I thought, what did he say to you? What? Again, not lost me. I had to focus a lot. Do you...
Yes, is the short answer to that question.
Thank you.
It can be difficult to take some complex ideas and then to discuss them. But I think one of the key is that all of these complex ideas that we work on are all built from lots of simpler, more fundamental ideas, which are easier to construct. So in 3 minutes, there's a bit that I have to miss out, unfortunately. If I was here to talk to you for 6 minutes, I could add a bit more of that detail and we could build it up to a more entire picture. If we were here for 9 minutes, we could make it even bigger and I could perhaps describe to you the entirety of how an organic solar cell or an organic TV could work. But in just that short amount of time, I wanted to pick one thing, which was how energy moves in an exciton, and relate that between a plant and future technology.
Nice pitch for Famelab, the director's cut there at the end. Ready to exit on cue. Please thank our nanotechnologist, Robert Thompson. Thank you. 5 down, 5 to go, which means we're exactly halfway. I won't go into the calculations, just take my word for it. 6th up, as selected by random, is Bert Rotten. As it says in his Twitter profile, I used to be a forklift driver, but nowadays I'm a doctor in cardiovascular biology. I hope to inspire people. Not that there's anything wrong with being a forklift driver. Now based at Cambridgeshire life science measurement and testing company LGC, Bert describes his work as developing ultra-sensitive immunoassays that can detect 0.00000000000000001 grams of protein in 1 litre of blood. I think that's a femtogram, but I wouldn't bet on it. I bet somebody in the audience will know. Now, Bert is getting married in a couple of months. He says he plans to teach a science class during his honeymoon, although you can take that in several different ways, and that his fiancée signed him up to Famelab, possibly to distract him from the wedding preparations. Whatever the reason, clearly worked. Stand by to be inspired and possibly uplifted by forklifter turned Famelabber, Bert Rotten.
Ladies and gentlemen, welcome to the very first boxing match in Famelab history. In the left corner, with the big smile, is the contender of tonight, Mr. Confidence. In the other corner, wearing the sad face, is the champion of the world, Mr. Insecurity. Well, okay, this is not me just doing silly, because we all fight with feelings of insecurity. When I was 16, I felt insecure because one of my teachers told me that I couldn't become a doctor. Nowadays, I have a PhD in cardiovascular biology. So tonight, I would like to give you 37 trillion reasons why we are all winners. Are you ready for that? So, the very first step. Are you ready for the very first step? Yeah. I said, are you ready? Yeah. Great. The very first step is the fusion of an egg cell and a sperm cell and the creation of what scientists call a totipotent stem cell. Now, a totipotent stem cell is like a factory assembly line because it can produce an unlimited number of new cells. The production of each new cell is controlled by a cell cycle checkpoint. Now, a cell cycle checkpoint is like a traffic light. When it is on red, it forces abnormal cells to self-destruct in a process called apoptosis. But when it is on green, it allows healthy cells to divide in a process called mitosis. Now, all these cells must pass the checkpoint, must pass checkpoint tests. But once they pass the checkpoint test, then they branch out. They branch out into those biological telephone lines that we call nerve cells. And they branch out into those complex memory sticks that we call brain cells. And ultimately, they branch out into a human being consisting of 37 trillion cells. Now, I don't know how many cells fail the checkpoint test, but I do know that we take it for granted that we go from a single cell into trillions of cells. So if there's one thing that I want you to take home tonight, then it is next time when that annoying Mr. Insecurity shows up, knock him out by remembering that there are 37 trillion reasons why we are all winners on this weird, wonderful, wacky, wonky, amazing planet. Thank you. Thank you. Thank you.
Thank you very much. That was very entertaining.
Thank you.
Very, very good. I wanted to pick up something that you were talking to our last contestant about, and it's about filtering down very, very complex science into analogies. I work with analogies a lot. How far do analogies get you in being able to fully describe your subject? Or do you just end up drilling down further into analogy?
Analogies are, you can't do them good and you can't do them wrong. It's really difficult. So you simplify things, and you don't want to oversimplify, because science is complicated and we should not talk like everything is really easy to explain. But analogies do help to make people understand and to give them that moment. And that's why I like analogies, because it really helps them to understand it. There is a thin line that we're walking here then.
Do you get frustrated though? Do you go, well, the analogy only gets me so far because I can't.
I did a PhD. The analogies don't frustrate me anymore.
You should come to where I am. It's all just analogy. It's great.
This idea of a checkpoint in cellular division and apoptosis, do those kind of ideas translate to something probably people think more about, like cancer development? And
that's a really good question, because exactly in the disease cancer, checkpoints play a crucial role. And what cancer cells do is they overrule these checkpoints, and they figure out ways to put all the traffic lights on green. So there are actually 3 checkpoints, and my favorite one is the last one, which is the division checkpoint. This is where the cell decides that all the things that it produced are okay, and that the cell is allowed to continue down the road of cell development. Well, I see this as that checkpoint is the cellular graduation party of the University of Life, where we all graduate and become PhD in cellular perfection. That's really true. And that's it is amazing. These checkpoints are awesome. And that's,
you lost for words.
Yeah, a little bit. Right.
Okay.
Very rarely lost for words, you'll understand. You're very confident. Has that come from doing science, or has that... It seems like it might have always been there, to be honest. Has that helped you be a scientist, being as confident as you are?
My confidence is because I learned a couple of things along the way that as Quentin introduced me, I started out as a forklift driver, and a lot of people told me that I couldn't become a scientist, until I heard a guy called Les Brown say that someone's opinion of you does not have to become your reality. And that really triggered something in me. And that was that I said, you know what, I can go back to university and get a degree, which I did. And it gives you a certain level of confidence when you overcame these obstacles. So to bring it back to the topic, if you think about all the obstacles that cells overcame, and there are 37 trillion cells.
Nice.
It is amazing to...
Nice segue. sorry. I'm confident your opinion of Bert Rotten will be excellent. Thank you, Bert. I also noticed from what he sent in that Bert is also very keen on Thai boxing and building his own furniture. If he doesn't win tonight, I wouldn't like to be his shelving. I also feel for him because of all the questions you could possibly have anticipated being asked about his presentation. Trying to defend the utility of analogies could not have been top of his list. Right, seventhly, we come to our Cardiff champion, or Welsh winner if you want to big her up, and she is Amy Jenkins. Asked to describe herself in a sentence, Amy opted for passionate, hard-working and devoted.
So hardworking, she has 2 master's degrees, one in sports psychology, one in neuropsychology, and is now studying for her PhD in subjective cognitive impairment at Swansea University, with the ultimate ambition of helping diagnose Alzheimer's not just weeks, months, or years earlier, but over a decade sooner than we can currently identify it. It is a bold, long-term goal. Right now, though, Amy's sights are set on something rather more immediate, the next 180 seconds. Her time starts just after your clapping stops for Amy Jenkins.
Look at me. I wonder if you are all seeing the same thing as each other right now. Whether you can see one or 2 of me. Whether you can see that I'm quite tall and that I have long blonde hair. Our eyes take in a lot of information from our surrounding environment, which is then combined with previously stored information we have about the world, not all of which reaches our brain, which is a good job well done by the brain, because we could never process it all. I couldn't guarantee that I could find a shadow of myself on this stage this evening. So here's one I made earlier. This is my shadow. I don't pay attention to it, it's just there. I know my shadow is there because my body has come in between the beams of light and the surface of the floor. I know that my shadow is attached to me, it comes with me everywhere I prop. Seems quite simple for the most of us. But for somebody like Mr. Thompson, who I used to work with, well, he had Alzheimer's disease. He had trouble with vision and perception. So he had trouble seeing things like shadows. He couldn't tell the difference between a shadow being a shadow and it being a physical object. He would quite often try and step over his shadow because he thought it was a hole in the floor. Let's think about the impact that this could have on Mr. Thompson's day-to-day life in his care home. Well, every time he tried to step over his shadow, he was much more likely to fall. Falls are a common and serious health problem and can have devastating consequences, especially for someone with dementia. But shadows are everywhere. Let's transfer some scientific knowledge into clinical context. Mr. Thompson's care home were advised to improve their lighting. They were advised to draw the curtains when low light was flooding in order to minimise the amount of shadows produced. Look at me again. Just imagine the confusion if you thought that my shadow was a deep, dark, black hole. Confusion that can be minimised by simple interventions like lighting. Now, we know all of this, but knowing it is just not enough. We need to transfer scientific knowledge into dementia-related environments in order to help them lead the best life that they can. Thank you. Thank you.
Wow. So in terms of the clinical setting, okay, some of these effects and impacts can obviously be translated. Can they also be used in diagnosis, some of these concepts that you're understanding about visual interactions with different species?
They certainly can, yes, because things like vision and perception, well, for instance, shadows are normally about mid to late disease.
Okay.
But we are looking at, like my area is trying to identify and characterise Alzheimer's disease about 10 to 12 years before diagnosis. So things like visual and perceptual difficulties, well, what it's done for us is to be able to help us identify that it's not just in those areas in the temporal lobe that dementia actually starts. It does start there, but what we found with looking at symptoms such as these, that we're able to look at different areas, like in the occipital lobe. So when scanning, they can actually see that there are quite subtle changes in what's called the extrastriatal cortex at the back of the brain. So we're able to see that there are changes in those areas as well. which can help towards...
So these are still quite complicated tests at the moment. Can you see a point where there are more simple tests that GPs could start to think about using?
Not so much with GPs as such at the moment. In care homes, if it's just that somebody's elderly within the care home and they start noticing things, because it's quite common in care homes or anywhere actually, where they think about this period of time in the evening, for example with shadows, that they get quite agitated. They can't explain what's actually going on at that moment for them, but they can, through anecdotal reports, yes, they can say, okay, something's not right here. It's either their vision, okay, let's check out if their glasses are okay and things like that. But otherwise, yes, they can use, most definitely. So, thank you.
Thank you for your talk. The link, you're at the interface between academic research and clinical management.
Yes.
How fun is that interface? What's it like to work there?
What now, from my research?
From your perspective, you straddle the line between academic research, sat in a lab, to actually rolling that out and actually getting it across Wales, across the UK, across the NHS. Yeah. How feasible is that? How easy is that? How frustrating is that? How fun is that?
It's very difficult, actually, because you try to be... like, I've worked clinically for a few years before coming back into academia. So it is quite challenging when you're supposed to be back in the office, doing all your reading, doing your research, and then thinking, right, it is quite complicated then to think, right, how can this work in the clinical context? How could people actually use this information? It is very difficult, and it can be very frustrating as well, not just from my perspective. It can also be from the clinician's perspective as well. And for within care homes, you know, we put a lot of pressure on the care staff as it is, let alone thinking, watch when they walk in past shadows. Yeah, and it's even more frustrating when it comes to money, because the simplest of interventions, like lighting or changing a toilet seat colour, for God's sake, it doesn't cost much, but it causes, it helps so many problems, changing a white toilet seat to a red toilet seat so they can see the toilet. You know, simple. So yes, it's frustrating because of money mainly as well.
Okay, we're not doing toilet seat research, shedding a light on the shadow world of Alzheimer's. Amy Jenkins and the cello. What she's going to find to do with that large cutout of herself after Famelab. Right, anti-penultimately, we have Jason Taylor. Call me Jace, he says, so we will. Jace is an algorithms engineer at Sharp Research Labs in Oxford, working on algorithms to make smartphones even smarter, yet even easier to use in ever more interactive ways, or at least that's the idea. And we're not doing that. He's learning to play the trumpet. So, he's got algorithms, he's got music, who could ask for anything more? Well, Jace can. He came along to see Famelab at Oxford and enjoyed it as an audience member so much, I thought I'd have a go the following year. And look where it's got him, all the way to the UK Final. Buckle up for a journey into Jace with Jace Taylor.
Right, we're going to do some science right now. I want you to get into pairs, or threes will work. As long as you've got someone you can play, go ahead now. What we're going to do is play 2 games of rock, paper, scissors. I'm going to say 1, 2, 3, show, and then again. Everybody ready? Found a partner? Ready? 1, 2, 3, show. 1, 2, 3, show. Now then, think for a moment. If you won the first game, did you stick with your item, or did you change it? What about if you lost or drew? What you did right then was a little bit of game theory in your head. Game theory is the science of decision-making in a competitive situation. You observed how your actions in the first game shaped your success, and then devised a strategy to be as successful as possible in future games. There's actually a secret trick to this game, which you might figure out by the end. Now, about the same time as the math behind game theory was first being formalized, there were murmurs of an awkward problem in biology. Behavioral biologists couldn't figure out why animals were surprisingly gentlemanly or ladylike when competing over resources. Now, you might not initially think this is the case, especially if you've seen the bickering in my house over the last after 8 at Christmas. But at least Uncle Frank gets to go home alive at the end of the argument. Oh, no. So sorry. And the reason was that human beings, like many animals, have evolved a sense of altruism or generosity. Rather than always taking everything for ourselves, sometimes we let someone else have that last chocolate, right? And maybe we do it thinking that one good turn deserves another, that, you know, Uncle Frank will give us his last Yorkshire pudding in return later on. But whatever the reasons, excuse me, whatever the reasons, oh, nuts. I'm so sorry. My last piece was about long-term memory, would you believe it? Whatever the reasons, this apparently unexpected behaviour has triumphed because it takes into account the other individuals' strategies as well as what we're planning. That's the real lesson. That the best strategies, as found in evolution, are the ones that take into account our competitors as well. And we can apply this lesson straight back to the rock, paper, scissors we played at the start. Research has shown us that normal human beings tend, in general, to stick with a winning item, to downgrade a losing item, and to upgrade an item that draws from one game to the next. But now that we know that, we can formulate our strategy to beat it. So, when you win, in the next game, play the item your opponent just played. When you lose, downgrade their item and play that. And when you draw, upgrade their item and use that in the next game. But whatever you do, keep it to yourself. Thank you. Sorry. Oh.
Never send Uncle Frank to our house. We would not be that generous with him. Good talk, thank you. Are there many examples where we take, where we can manipulate human behaviour? First of all, presume this is used throughout marketing and throughout business. This sort of understanding.
It's used in that way, yes, but it certainly appears in all kinds of fields: economics, gambling, dating, all sorts. But yes.
Are there many instances where we look at the animal kingdom and go, brilliant, right, let's use that, manipulate that, let's work on it?
Certainly, yes. So there are lots of cases where this behaviour sort of naturally occurs in the animal kingdom. You probably know more about this than I do, but vervet monkeys, for example, are a case where they live in social groups, and one of them or a couple of them are posted as lookouts. And then when a predator approaches, those lookouts start screeching, even though it actually draws the attention of the predator to the scout. It makes it more dangerous for the scout, but they're relying on the idea that you know, the rest of the group will protect them, and then one day they won't be on duty and they'll still benefit from the protection. So there are, you know, you can take models from things like that. But it transfers this kind of concept of cooperation and how it naturally emerges can be applied in all sorts of situations, like dating. There's a really great one, if you'll indulge me. There's a really great example in the film A Beautiful Mind, if you've seen it, about the mathematician John Nash, who was actually a big player in this field. And him and his friends, they go to a bar, and they meet this huge group of beautiful women, and they're all quite hopeful. They're hoping to get a dance and maybe some dates or phone numbers at the end. Well, there's one particular, really, really beautiful, it's a bit of an objectifying example, I apologise, ladies. But there's one particularly beautiful lady, the star, let's call her, and all the guys think, oh, I definitely want to try and get the phone number from this one girl, this star. So what they think, you know, they're all going to go for this girl, and then inevitably, at best, only one of them will get her, and then the rest of the guys will have to go to the other women who are equally lovely. But because quite rightly they're now the second choice, no, I'm not interested in being second choice, so everyone else goes home. But if we take a leaf out of this cooperative behaviour book, and all of the men ignore the starlet and go for different ones of the other group of equally lovely women, everyone stands a much greater chance of getting a date and a phone number, except the starlet. And some of us are lucky enough to marry the star. But in general, that behaviour benefits everybody. So that, you know, this is behaviour that you can directly take from animal cooperation.
So my daughter, who's 9, has a version of rock, paper, scissors with her friends, which is rock, paper, scissors, black hole.
Ooh, I like this.
Where black hole clearly beats everything.
Clearly.
But they don't always choose black hole.
Tactical error.
So is there... You would think so. You would
Always
think so.
black
Well,
hole.
if I play with them, I always win. But the question is there any analogy in the natural world where something that could always win would choose not to?
Apart from an actual black hole.
Apart from natural
yes.
black holes. Is that a sense of altruism even ever seen in systems where there is something that can always win?
Yes, in a sense. And it's to do with how stable these behaviours are. So when we talk of evolutionary game theory, we talk about how stable these behaviours are as strategies, how long they last from generation to generation. And strategies like this, you know, like the one we described in rock, paper, scissors, they tend to exist quite harmoniously with other strategies like just throw a random symbol out there, whatever it may be. But then every now and then you get call it a mutant or an invasive strategy. It might be suddenly growing enormous fang teeth or something like this, or a terrible poison that suddenly comes along and for a while sweeps everything off the board, just wins and wins and wins. But the problem with those strategies is actually, this is the real current area of research in this field, called zero determinant strategies, which is some matrix maths for another night. But these things, they swoop in and are very, very effective for a short term. But as soon as other strategies cotton on to what's going on, hang on, this guy's got massive teeth. What I need is some armor, or this guy's got terrible poison. What I need is the natural antidote in my mouth. And they tend to be, they're what we call evolutionary unstable. So those things will sweep in and be very fantastic, but then they'll be overcome by another strategy, or they'll just die away because they're not robust and reliable. So in the long term, these more stable strategies, like the one we discussed, will be around for longer.
Quentin, you're like a predator.
Yes.
You're like some teeth.
And the data tips. Okay, very quickly. 1, 2, 3. 1, 2,
1, 2,
3.
3, show.
1, 2, 3.
Show.
Yes. But will he be a winner later? One more time, please, for Rock,
Thank you.
Paper, Scissors. James Taylor. I'm a time predator, Dan. So
I know, but I've got
got
questions. That was interesting, and you
It was,
interrupted.
but he's only allowed 3 minutes. That's the way it goes.
I know, but
We have this thing called the bar later. I don't know if explained.
okay.
Rather than trying to chat up the women there, following his technique, you can chat to him then as well. Right. And if anyone, by the way, having seen Jace, would like to follow his example and enter Famelab next year, either because you think it's wonderful or because you think you can do better, then the details are available via famelab.org. Right, last but one, we have the Bad Lizard, or at least the Bad Lizard is his Twitter handle. His less convincing real-world identity is Chris Basu, PhD student at the Royal Veterinary College in London. Further evidence of his split personality are that he tweets things like, "Thank you, old me, for taking decent photos of dissections early in your PhD. Current me is amazed you even knew what you were doing." Chris and/or the Bad Lizard like baking, fossil hunting, peanut butter, and large land mammals. His sticky giraffe and ammonite pie is apparently to die for. Chris is hoping his research on giraffe anatomy and biomechanics could help to improve the treatment of giraffes kept in zoos. It's something he may or may not talk about in the next 3 minutes. I wouldn't like to stick my neck out. Stick your hands up, though, for Bad Lizard, Chris Basu.
This is a story of triumph, tragedy, and evolution. We start in the Kruger National Park, South Africa. A giraffe is being chased by two lions over jagged ground. The giraffe stumbles, comes crashing down, and the lions move in. Now, the tragedy for this giraffe is also linked to one of its greatest successes. Giraffes triumph with their ability to get the food that nothing else can reach. But a long neck means long nerves. A long neck comes with a catch. Right now, if we look at a human, the nerves in the soles of my feet are firing impulses to my brain. And they're traveling through my body at 50 meters per second, taking 0.03 seconds to make the distance. That's 10 times faster than I can blink. And in you and I, we usually cope fine with these small nerve delays. Even if I trip, there's enough time for the pressure sensors in my feet to tell my brain something's wrong, for my brain to go, help, and then to send back a response to my foot to correct the problem. Fine. But what about the giraffe with its supersized neck and its long legs? Well, that distance from head to foot in a giraffe is slightly longer. To measure it, we need to examine one. Back in the Kruger, the lions are busy eating our giraffe. If we just shoo them off, shoo, shoo, we can take a closer look at what they've left behind. Over here, the head. When the brain sends a nerve signal from the giraffe's head here, it has to travel down 2 meters of neck, and then along another meter of back, and then down 1.5 meters of back leg just to get to the back foot. And that is a huge distance. It's 5 meters. And it's here that we hit a snag. You see, the speed of nerve conduction in giraffes is roughly the same as what it is in humans. This means that nerve delays in giraffes are much longer. When a giraffe stumbles, the time it takes from sensing the problem to sorting it out is close to 1/4 of a second. For a giraffe galloping for its life away from lions, 1/4 of a second is more than enough time to trip, fall, and get eaten. The lions, when they chased our giraffe over uneven ground, they managed to exploit this evolutionary trade-off between triumph and tragedy. But you'd have thought, in the 2 million years that giraffes have been around, maybe they would have sorted out this problem. But evolution is not perfect. It's messy. While giraffes enjoy the perk of eating from the tallest trees, this perk comes with a price. With evolution, there is no such thing as a free lunch. Thanks.
Thank you very much. It was a lovely story. It was, again, reminiscent of a Just So story.
Yes,
How the
yeah,
giraffe
quite.
its neck and Lamarckism and such things. Can you just explain a little bit about your research and what it is that you are actually working on, what it is you're actually doing?
Yeah. So I'm interested in these trade-off questions because Just So stories, you know, it's so common we hear things like, oh, this is perfectly evolved for this, you know, this is perfectly adapted for that. And it's a nice Just So story, but it's not the whole story. So there's always trade-offs. You know, if you get good at one thing, inevitably you sometimes get worse at something else. And giraffes are a great example of this because they're large mammals. They're related to lots and lots of things that we know, but they're so strange. You know, they're so derived. They've got such odd limb proportions. So I'm looking at how we can study anatomy, and relate that to function. And giraffes, they're a great example because there's loads of them in the UK. We can travel to zoos and look at them. And we can do things like look at their anatomy through dissection or through imaging, like CT scans. And we can do more fancy things like computer modeling, where we actually take a computer giraffe, because it's very difficult to do experiments on giraffes, and we can actually do experiments on the computer model instead. And from that, we can learn these kind of lessons.
Well, what are you actually trying to learn? What are you looking for?
So I want to know, why do giraffes walk and run in such a peculiar manner? They're a little bit strange. I want to know things like, because they have really, really long legs, does that mean their muscles are at an advantage or a disadvantage? So I'm looking at these, these kind of trade-offs.
How do we know how fast nerve signals are transmitted?
Yes, it's a great question. People have looked at nerve speed in lots and lots and lots of animals. In giraffes, it's done when giraffes are actually asleep. So the particular study that this comes from looked at the speed of the sciatic nerve. So that's a nerve that goes from here to up here. They can basically give the nerve like a little electrical flick, a stimulus. And they can measure the time it takes from that stimulus to go through what's called a reflex arc. So that's like your patellar kneecap reflex. And they measure the time that they get a response. So that's the actual work that they did. And there's a little bit of extrapolation and assumptions that go into, how long does it actually take from, to get to here, to the brain.
And that's the same for virtually all mammals?
Yeah, it's really weird. If you look at animals like a mouse, and you compare it to an animal like a giraffe, and look at all the animals in between, they actually have relatively the same nerve conduction velocity. So you'd have thought, well, if we're going to get bigger, we have to speed everything up, to maintain these nerve delays. But no, it doesn't work like that.
Thank you for your talk. Are you part of John Hutchinson's group?
Yes.
Good.
Yes.
That was my question. No, are there any, I know John's very keen on looking at the stuff that his group works with and either relates that to from extant to extinct species or links it to welfare standards. Is there any remit or any room for your work to look at extinct mammals or titanosaurs or animal husbandry? Is there any link for that?
All of the above. Which do you want?
All of it. He's miles away yet. He's got a touch on his shoulder before he stops talking.
I don't want to lurk in a predatory way in case he trips and I have to eat him. So.
He is quite tall.
Yes, to answer one of those questions, giraffes have this wonderful, rich evolutionary history. They have evolved from relatively short-legged, short-necked animals to the long-legged animal that you see today. And with that shift in these proportions, we should see these exact kind of trade-offs that I've just been mentioning. We can use this kind of research to actually try and benefit giraffes living today, living now. It's quite common in zoo settings that large animals tend to suffer from various kinds of foot disease. And there's a lot we just don't understand about these animals. And this kind of research, so knowing how anatomy relates to function, knowing the kinds of forces that go through the legs, knowing the strains that go through tissues, can actually help us to work out why these animals get various kinds of foot diseases.
Okay, Tripping the Light, fantastic. Please thank one more time Chris Basu. And I am mostly giving the anagrams a night off tonight, but I can't resist one because I noticed Chris Basu, I crash bus. And finally, our final finalist, although statistically unlikely to be our finest finalist, is Kyle Evans. Now, Kyle is the middle bit of a Venn diagram that has mathematics on one side and folk music on the other. A maths teacher in Hampshire, he's also spent the last decade touring the country in various indie folk bands. Kyle's recently released a new online EP, Songs Written in Head While Cycling, which he describes as 5 simple songs about life, etc. 1 country song that a guy promised me 50 quid for and never paid up. He says that one of several reasons he's really enjoyed being part of Famelab is that, unlike at some music gigs, it's nice to play to an appreciative audience. So please show some appreciation now, preemptively, for Kyle Evans.
This here is my favourite book. It's called Flatland by Edwin Abbott Abbott. And we'll just get this cleared up nice and early on. His middle name and surname were both Abbott. This was written 100 years before I was born, but I like it so much that we named our son after the author, actually. And it describes Flatland. Now, Flatland's an entirely 2-dimensional universe. So if you can imagine a whole world that's like a giant sheet of paper. And the inhabitants of Flatland are 2-dimensional creatures. And they go about their daily life in a world where there's length and breadth, but there's no height. There's alongside and in front of, but there's no above or over. Now, the maths in Flatland is fairly straightforward. It's the maths you remember from school, what we'd call 2D Euclidean geometry. But the pop music in Flatland is quite strange. Here's an example of pop in Flatland. Like a road next to troubled water, I will bend around. But it goes on to describe Lineland. Now, Lineland is a whole universe, if you can imagine it, that's arranged along a straight line. And the inhabitants of Lineland are 1-dimensional points and tiny line segments, and they move along this great line. And if 2 of them come head-to-head, they've got no way of changing order, because that would involve either going above or below, which we've ruled out, or going around the other one. There isn't even a concept of around in a 1-dimensional universe. This means you spend your whole life just bumping into the same 2 people. Now, I live in the country, so I know sort of what that's like, but for most of you, that would probably feel quite strange, I expect. And again, it does quite strange things to pop music. I feel it in my vertex, I feel it in my node. Your love is in front of me, nowhere else I can go. But the point of this, the point of the book is that just as the Linelander could never understand Flatland, and a Flatlander just couldn't comprehend our 3-dimensional universe, so we humans struggle with the 4th dimension, don't we? What is the 4th dimension? What does it look like? And this is when it's great to be a mathematician, because mathematicians don't really care what the 4th dimension looks like. Just as long as the maths works and the numbers hold, we say, yeah, 4th dimension exists. And the same goes for the 5th, 6th, 8th, 10th, 100th dimension. Just as long as the maths works and the numbers hold, it's useful to us. And indeed, working in more dimensions than we could ever see has led to some of the great proofs of recent years, like Fermat's last theorem and the Riemann hypothesis. So when Edwin Evans turns to me one day, says, Daddy, is the 4th dimension real? I'll plant upon his head a kiss and say, son, just like £1,000,000 are that old Father Christmas. Just because you've never seen it doesn't mean it don't exist.
Have you written some 10-dimensional?
10 dimensions.
What would that sound like?
Give me can I start with 4 or sort of 5 dimensions?
All right, then. Yeah.
4 is the magic number. A new dimension to add to, alongside, in front of, or under. Sorry.
I thought you were just going to add dancing then.
I thought I was going to get freestyle.
Yeah.
So it was pretty freestyle.
It was pretty freestyle. It's pretty good. Does this kind of analogy, when you're teaching maths, all right, so you have to think about this a lot. Is there a difference, do you find, in trying to take maths examples to 11, 12, 15, 16-year-olds? How far can these analogies stretch from an audience like this to a classroom for students?
Well, I think in mathematics, just like every other subject, creativity is really important, and a lot of students, when they're taught maths, it's very, it can be, if not done properly, these are the rigid rules you must adhere to. Someone's worked it out, now you just do something simpler. But students should be encouraged to think outside the box, or cube, should I say. But there's no harm in it, It's good for students to see, we're never going to be able to see what the 4th dimension looks like. It doesn't mean that we can't make calculations and it can't be useful to us.
Very good talk, thank you. As an aside, I'm very bad with who said what when, so I write little ways of remembering who's been speaking. I've written Chris Packham with a guitar. I'm so sorry. You've just said what's like Packham. It's hilarious. I had to say that.
Now, thank you.
Linking on from Mary's question, how important is it that the general population has an appreciation for maths? I know it's when you go to a shop, it's very important, but how important is it that I love maths? Or is it important that I love maths? Because I don't.
Yes, you do.
Oh, you
do. No,
don't.
You just don't know it.
No, I mean, I understand. It would be what kind of, if your hobby was something that everyone loved, no one, sometimes it's nice to have something that not everyone loves. But, I mean, you're right, there's sort of different types of maths. There's the maths that is useful to you when you get your bills or you go to the shop, and then there's high concept mathematics, which you can pick up and take an interest in, regardless of where you stopped your mathematical studies. If you're thinking about paradoxes or logic problems, the mathematics behind it's really interesting. But if you just like the logic puzzle for what it is, that's fine also.
Thank you.
Carl, I just have to. You didn't really call your son Abbott, did you?
No, just the Edwin.
Just
not
the Edwin. Just thought we'd check on that one as well.
Evans, no.
One more time, please. No encores, though, for Kyle Evans. Favourite book of mine, too. Now, that was the final track on Now That's What I Call Famelab 2016. From the millions who could have entered this year and the thousands, well, hundreds who actually did, we have whittled and winnowed it down to just these 10. Now, we could just leave it there, but as a contest, that would be quite rubbish. So, Famelabulous as they all were, this is where we make days and break hearts. The judges need to be on their ways to swiftly decide on one winner and 2 joint runners-up, and they need to do it as fast as possible. So can we speed them on away, please, with some applause? Last chance to shout for anybody you want to win. That's it, influence them in some subconscious way. Now, while they are away, we have work to do, the audience prize. Now, I approach this rather nervously. There are 2 parallel histories of Famelab. One is a long and glorious tradition of encouraging and helping develop science communicators who are brilliant and have gone around the world. The other is of large numbers of supposedly error-proof voting systems for audiences that have gone wrong. Down the years, we've had several mishaps, including one early one at Cheltenham where I was stranded on stage for 40 minutes while we tried to get the thing to work. If you think it was bad for me, Think how it was for the poor audience. Right. Now, I mention this because after several years where the voting has gone without a hitch, tonight we're trying something new. Oh, God. It'll probably be fine, but I'm just trying to raise hopes raise kind of lower hopes and raise expectations. Anyway, just get you worried. So here's what you do. First and most importantly, decide which of our top 10 you think was number one. I implore you, ladies and gentlemen, to put aside emotions and just focus on logic and the overwhelming evidence in reaching your verdict. Sorry, I've just been watching OJ, The People versus OJ, and it's had too much of an influence on me. If you can, resist the urge to vote for someone just because you know them or came with them or you're from the same area as them. The voting is anonymous, so even if you promise to vote for your friend, girlfriend, partner, wife, whatever, they will never know. Vote for who you think was best. Secondly, get out your smartphones or other internet-enabled device. And you have two options here. One is, as it says, you can try and hook up to the cloud and enter the details. If not, make sure your network is selected to ignore the cloud and just use the existing coverage. If you do not have an internet-enabled device, congratulations, you have just been disenfranchised. And it could be, of course, that everybody's disenfranchised if you can't get the Wi-Fi in the system to work here. So are people managing to find some kind of signal? Good. Now, once you've done that, go to, thank you very much indeed, as it says there, live.voxvote.com and enter this PIN number, 66059. As I'm sure you've spotted, that's the sum of two primes, 7 and 9,437. Sure you all knew that. So enter that. Now, if all's gone well through the miracle of technology, or just an old-fashioned miracle, you should be able to vote for whichever of the 10 Famelabbers you think was best. As soon as you've voted, you can click to leave the event, and I will give you a couple of minutes, and then the vote will be closed anyway. Are people getting through, sort of? Yeah, obviously I don't expect everyone to say yes, but some of you are. Okay, this is probably going to be, as we say in the trade, somewhat haphazard. Not everyone may get to vote, but it should be done on a random basis. Okay, you've got one more minute, I think. As I say, once you've voted, leave the event, do not go back. So, for instance, the three people there with the Go, Amy, Go poster, you're not obligated to vote for Amy. Just whoever you fancy. They are obligated. Oh, sorry, Amy, I should have checked that. Okay, can you make a kind of low murmuring noise if you've successfully voted or appear to have? Excellent. Okay, we're going to give you 30 more seconds and then it's done. Okay, last few, counting down now. Brilliant, thank you. Now the voting is done. The result is, I think, already in through the miracle of technology, but for the sake of drama, we are going to keep it until afterwards. When we come back, we're going to keep it and the bottle of champagne for our winner on ice until we're back with the judges later. While they are reaching their verdict, however, we have got a mystery to solve. The mystery of how the best-selling novelist of all time, Agatha Christie, poisoned so many of her characters using so many different compounds. I am delighted to be able to hand over the stage to chemist and author of A is for Arsenic, The Poisons of Agatha Christie, the intoxicating, with the emphasis on the toxic, Dr. Catherine Harkup.
Hello. I'm going to tell you how to kill people. Please don't kill people. Feel free to take notes, but anything you write down can and will be used in evidence, etc., etc. If you want to kill people, basically your best go-to guide is Agatha Christie. She's extremely good at it, particularly when it comes to poisons. She killed more characters more often and more accurately with poisons than any other author. She killed over 100 characters with over 30 different compounds. Now, the judges going to take some time, but we're not going to get through 30 compounds, so I'm going to pick one. I'm going to talk about strychnine. Strychnine is a bit of a favourite of Agatha Christie. She killed 7 people with it, obviously fictionally, and she used it in one of her most famous novels, her first novel, The Mysterious Affair at Styles. She wrote this in 1920 when she was working as a dispenser in a hospital chemist dispensary. So she knew her stuff, and she really went to town on her chemical knowledge in this book. In case you haven't read it, I'm not going to give out spoilers, but in case you haven't read it, this is the kind of setup. You have a big country house where, let's face it, all the best murders take place. And you have a very rich older woman with a very poor younger generation. And just to make sure that this rich old lady is about to die, she has just married a much younger man. So her time on this earth is limited. One evening, the whole household is woken up as this old lady, Mrs. Inglethorpe, is having horrific convulsions. And everyone storms into her bedroom and they witness these horrible throes of agony she's having on her bed. Coincidentally, Captain Hastings just happens to be staying in the house. And Captain Hastings, who, God love him, is not the sharpest tool in the box, even Captain Hastings recognizes that this lady has been poisoned with strychnine. So I'm sure you're thinking, what is strychnine? How do I get hold of it? What you need to do is you need to go to India, because strychnine comes from a plant called Strychnus nux vomica, which grows in India and other regions around there. You are looking for a kind of shrub with beautiful orange fruits. The poison is not throughout the whole plant. You need to get the seeds from the fruit, and they're flat, disc-like seeds. They're quite big. They're about the size of an old 5-pence piece. They've got a tough outer coating, so you're really going to have to grind them up before you extract the poison. Now, this is frankly quite a lot of hard work. Much easier in 1920 when Agatha Christie was writing, when strychnine was frankly everywhere in the house. It was used to kill vermin. There's a blue bottle in a drawer upstairs that may or may not contain strychnine. Strychnine itself is white colourless crystals. There are white colourless crystals on a tea tray. Is it sugar? Is it strychnine? Who knows? There is also strychnine on Mrs. Inglethorpe's, our victim's, bedside table. Why on earth would you have strychnine on your bedside table? Well, it's because a doctor has prescribed strychnine to this little old lady as a tonic. The idea was that the bitter taste of strychnine is the most bitter tasting substance known to man. If you're going to use it, disguise the flavor, okay? That's all I'm saying. The bitter taste was supposed to increase your appetite, and it was supposed to pep up your nerves. And oh boy, will strychnine pep up your nerves. Because what happens is, so nerve signals, as we've heard from our contestants earlier, electrical signals run along our nerves, but where 2 nerves join, or where a nerve joins a muscle, there are little chemical signals that are released to cross the gap and pass on the message. For our purposes, there are 2 chemicals involved, 2 that we're interested in. One is called acetylcholine, and that's the go message to the nerve or the muscle cell. The second compound is called glycine, and that's the stop message. Now, normally, both these chemicals are released at the same time, and it's the balance of these 2 chemicals that gives you a range of sensation, and it also means you don't get false triggers. What strychnine does is it blocks the glycine receptors and it switches them off. So all of the stop messages vanish. And all your nerve is receiving is go, fire, do it now, urgently. So, well, here's what you do. You get your strychnine and, as I said, disguise the flavor or ensure that your victim swallows it in one go. You need about 100 milligrams, really not very much. Then you've got about 15 minutes to get out of there and establish an alibi, because that's when symptoms are going to start to present themselves. You're going to start off with a bit of twitching, but this is going to progress to whole body convulsions. Now, the muscles on your back are generally speaking stronger than the muscles on your front. So victims arch up like a C shape with just their heels and the back of their head resting on the ground or the bed in the Mysterious Affair at Styles case. Christie is very, very good in her descriptions, very, very accurate. You have bouts of convulsions, and then you have periods of calm. And then something else will trigger your nerves, and you get another bout of convulsions. Most people survive 2 to 5 bouts, and they either die because of exhaustion, their muscles are literally gone into overdrive and they collapse, or most likely, the muscles that control your breathing become contorted and you can't breathe, you suffocate. Now, if you're not scared enough already, because strychnine is interacting with nerves, it also has a slight interaction with nerves in your brain. And it means that normally if you have convulsions, a person is unconscious. But in the case of strychnine, these people are horribly awake and horribly aware and hypersensitive to what is going on. This is an agonizing, prolonged death. If you're thinking cozy tweed and country houses for Agatha Christie, no, it's really quite horrific. TV has, I think, kind of promoted this myth of people, people sit down for a meal, they eat a bit of food, choke a bit, fall on the floor dead. No, this is not what happens. It takes ages. So in the case of the Mysterious Affair at Styles, we have our victim in these horrific contortions. But thankfully, because of Captain Hastings, he draws in the fabulous Hercule Poirot to sort out the mess. Because it's not just one strychnine, one source of strychnine, one compound that has killed Mrs. Inglethorpe. Not only do we have to figure out which source of strychnine has killed her, it's, in actual fact, there are three compounds to kill one person. Now, after eliminating all of these different sources of strychnine, it's not the strychnine used to kill the dog. It's not the strychnine from the blue bottle up in the drawer upstairs. It is, in fact, the old lady's own medicine that kills her. Now, people think, well, this is a time when people were making up medicines by hand. There was no pre-packaged material, and so people made mistakes. Maybe a mistake had been made when the tonic was made up. So what they would do to make the tonic, they would have about 100 milligrams of strychnine, a lethal dose, and they would put it into a bottle and they would dilute it. And then you take a spoonful or two a day as your tonic. So if the tonic was made up wrongly, you would expect Mrs. Inglethorpe to suffer on the first dose. But actually she died after the last dose. So clearly there wasn't an error that was made up in the tonic. So we come on to our second compound. This is how brilliant Christie is. She's a really good chemist. Also on Mrs. Inglethorpe's bedside table is something called bromide powders. These were the first kind of sedative drugs that were available, and you would use them basically as sleeping powders. The unfortunate thing was, if you added a bromide powder to your tonic bottle, a little chemical reaction caused the strychnine that was beautifully dissolved beforehand to turn into a solid and sediment or precipitate to the bottom of the bottle so that a whole dose was available in the last spoonful. Now, when Christie was training to become an apothecary's assistant, it is fabulously called, basically a dispenser, she would have read a book called The Art of Dispensing, and it very specifically cites a case where it says, do not mix bromides and strychnine because of this effect. A woman died taking the last fatal dose of her tonic. So it was a specific real-life case that Christie drew for her inspiration for this particular story. But I said there were three compounds. Well, compound number 3, this is where it gets even more complicated. Compound number 3 is a narcotic. Now, Christie doesn't tell us what narcotic, but from the symptoms we can guess what it was. She describes a delayed action of this strychnine. So I said you had 15 minutes to get out of the room. But actually, if you're clever, you can prolong that. Because she describes a delayed action in swallowing strychnine. Strychnine can only be absorbed from your small intestines. It cannot be absorbed from your stomach into the bloodstream. So if you have a way of delaying the contents of your stomach moving into your intestine, brilliant, you can delay the symptoms. Well, opiates, quite commonly, as well as knocking you out and getting you high and relieving pain, they also slow the muscles that control movement of food through your gut. So it means that heroin addicts and morphine addicts are quite often constipated. It also has the benefit, if you're trying to kill someone with strychnine, you can delay those effects for hours. So you can be well away doing something else completely with loads of people giving you an alibi. 3 compounds. Now, please don't do this. Don't know if I've mentioned this. Killing's not a good thing. There are consequences. You cannot get hold of strychnine in Europe today. It is considered too dangerous. It is not on general sale. It was, up until 2006, available to kill vermin. But they finally banned even that use in 2006 because it caused unnecessary suffering in the animals. So you are going to have to plan a trip to India if you want to do that. The other reason you shouldn't use strychnine is it's very, very detectable. It's very easy to kill people. It's almost impossible to get away with it. So, very detectable. It's been detectable since about 1850. Don't even bother. The other good thing, if you taste something, if you go home this evening and you taste a bitter drink and you worry about it, I mean, watch for twitching. And if you get a bit of I don't know, maybe your partner's been mentioning insurance policies. If you get a bit of twitching, call an ambulance, because there's actually quite a bit that you can do to alleviate these symptoms and save people. Thankfully, there are many, many antidotes. I was worried that the judges would come to a really quick decision and I wouldn't get to this bit. But there are antidotes. Because all of your muscles are contracted, very, very tense, what you need is something that will relax your muscles to counteract those effects. Fortunately, there are many, many muscle relaxants available. My favorite of which, it's particularly apt in this case because it comes from the same genus of plants as strychnine. It comes from Strychnos toxifera, but its common name, the poison that's extracted from this plant, is called curare, which you might have heard of if you read Sherlock Holmes or watch the film series. So curare is known as an arrow poison in South America. People tip their arrows with it and they shoot it at monkeys, whatever it is they're trying to kill. Curare interacts with those same glycine receptors on those nerves that I talked about before, and it sticks very, very tightly to that receptor, but it switches them on. So all the nerve hears, or the muscle, the only message it gets is the stop message. So all your muscles relax, which means that if you're hunting a monkey or whatever, you get floppy monkey falls out of tree, floppy monkey can't breathe anymore, so floppy monkey suffocates. You might think, well, hang on a minute, I'm going to eat that. So I'm going to eat. Curare has this brilliant property that if it gets into your bloodstream, you have this horrific paralyzing effect that you can eat almost as much curare as you like, and it will pass straight through you and be excreted with no effects whatsoever. So your floppy monkey is completely safe to eat. I collect weird and wonderful books. I can't go past a secondhand bookshop without going in. I found this book from 1910 that was chemicals used by med students, and it describes all these beautiful experiments that med students could carry out on brainless frogs. It even tells you how to de-brain your frog. It's a great book. And you get your brainless frog, and you can kind of stroke its back with a bit of strychnine. And frogs, their strong muscles are on their front. So you get frog on its chin and its toes. Tense frog. And then you stroke it with a bit of curare solution, and you have floppy frog. And then you give it a bit of strychnine, you have tense frog. And then you give it a bit of curare, and you have floppy frog. And you can go backwards and forwards until everyone gets bored. What's wonderful about poisons is that not only are some of them lethal in quite horrifying ways, but they're also antidotes to one another, and they do have medical applications. So curare is in use medically. Strychnine, no. No, that whole tense thing, the appetite thing, no. It's been decided that's not a good benefit. It's off the pharmacopoeia. Although occasionally I do this talk and little old ladies come up to me afterwards and they say, oh, I had double pneumonia once and they gave me strychnine to build up the muscles in my lungs. And it's like, oh, you're fine. You're still here. Well done, you. So strychnine has now completely disappeared from the British pharmacopoeia. Curare has stuck around. Because actually, if you can administer curare locally, obviously a refined form of it, they're not literally bargaining for the tips of arrows from South American Indians. They are, the refined compound is used particularly in abdominal surgery because your muscles are, even when you're unconscious, they're moving food through your gut the whole time. And if you can calm those or even stop them, then surgery becomes much easier and you can cut into those muscles, there's less tearing, you heal better, it's much better all round. So poisoning is all about the dose. Too much of anything is too much. Too much oxygen can kill you, too much water can kill you. It's about 7 litres. Your victim's going to notice because you're going to have to drink it quite quickly. So most people, when they're talking about poisoning people, they go for quite small amounts. And curare in quite a small amount, yes, it will paralyze the muscles, but it does, in appropriate doses, have a real benefit. So, I recommend to you The Mysterious Affair at Styles, because even though there is a lot of brilliant science about strychnine, you wouldn't think of Christie as a scientific writer. But she's a very good science communicator. She gives just enough information so that you get all the clues, so you think, yeah, I can work this out. I'll get there before Poirot. And I promise you, I have read every single one. Every single one. I guessed once. Once before Poirot got there. Or actually, I think it was Miss Marple. But anyway. So there's lots of information about that. There's very little information about forensics or antidotes. She's quite cruel, is our Agatha. Most people, if they're there, they're writhing in agony, they're going to have brandy shoved down their throat. I don't know what it was in 1920s and 1930s. Brandy was just the cure-all for everything. It doesn't work, okay? Please don't do that. Even if you don't happen to have a bit of curare lying about your house, or maybe even if the ambulance doesn't have it when they come to your door, there are other things you can do for your victim that won't make it, frankly, worse. In the case of strychnine, what you want to do is just remove all of those impulses, those triggers to the nerves. So basically, just keep everything quiet. Get your victim in a room, nice and dark, so there's no light triggers, nice and quiet, kind of stuff up all the doors so that there's nothing to set off these horrific contortions. And after that, it's fine. It genuinely does take hours. So it's very unpleasant hours, but it does take hours. There's a good chance of strychnine survival today. I should tell you about a real-life case, I suppose, because Christie often used real-life cases, and strychnine has a bit of a history as a bit of a choice for poisoners, usually medical poisoners, which is quite worrying because they would have known very precisely what was going to happen to their victims. One guy, Neil Thomas Cream, he was born in Scotland, but he spent most of his life in America. And after he'd served a prison sentence for killing one person with strychnine, he came over to the UK, and he turned up in London, actually, not that far from here, and he started killing prostitutes. This was 1892 that he started doing this, and he would meet these women, and he would offer them a pill. Remember, this strychnine is very, very bitter tasting. So he would offer them a pill. He'd say, this will cure your complexion. It will just clear up your complexion. You'll look wonderful. And they were like, oh, brilliant. Took the pill, and then he would hightail it out of there as fast as he could. And they died many hours later, writhing in agony. Killed 4 women. And as I say, in 1892, if anyone knows their history, this is round about the time of Jack the Ripper. Now, it is alleged that Neil Thomas Cream, he was caught, by the way. This is the moral. If you're taking inspiration from Agatha Christie or real-life cases, they get caught, okay? Don't use that as this is a how-to guide. No, because you know what? The police can read those same things, and they can figure it out too. They did actually find Neil Thomas Cream, and he was sentenced to be hanged. And it is said, as he was hanging, just before the bolt was drawn below him, he said, "I am Jack," and he fell through the trapdoor. It's not true. Sorry to spoil it. But it's a lovely story. Thank you very much for your time. Thank you.
And I think we should give Catherine an extra round of applause, because it's one thing to do strychnine on a strict schedule, but when you're having to film not knowing quite how long the judges are going to be, and you're killing off one more person and one more person and one more person, it's a tough thing to do. So one more round of applause, please, for Dr. Catherine Harker. Now, I should also say the full-length, multi-death version of that show can be seen at the Cheltenham Science Festival on the 9th of June, the very same day as the international final. So you can go and see Catherine's show first and come along. And isn't it a great thing about Famelab? You come for the future stars of science communication, and as an added bonus, we teach you all how to become mass murderers. So this is the point where we find out which of our 10 belladonnas and mandrakes are going to be winners and which are about to have their hopes cruelly poisoned. Can we get all 10 finalists back up on stage, please? Ooh. Just shuffle across as you see fit. Leave some room for the judges, that's fine. Brilliant, some have brought their fish, some haven't, fine. Now, before we get to the decision of our judges, the wisdom of crowds, we're going to start with the one that you did, the audience vote. Now, I can confirm, if you step forward, Catherine, that although Catherine has only just been handed this envelope, rather more importantly, she has only been shown the champagne in front of other people. She has not had a chance.
Sealed.
It is sealed. She has not touched what the contents are. So, Catherine, can you open the envelope and reveal the name of our audience winner?
Right, I'm going to put this down, otherwise there will be an accident of some description. So, let's have a look in here. The audience vote winner is Kyle Evans.
Oh!
Well done. Right, first of all, well done, sir.
Thank you very much.
Right, that one first.
Lovely.
The important one.
Thank you very much.
Congratulations, well done, sir.
Thank you, Catherine. That is either a tribute to Carl's brilliant genius or the fact that you guys have terribly short-term memory since he was on last as well. Now, can we get our judges back here as well, please? Preferably some well-earned applause. So how does the audience decision measure up to that of our unelected, hand-selected judges? Dallas, as our chair, can we start by revealing who our two joint runners-up are, each of whom get £200 cash plus £300 for approved public engagement activities? And perhaps first, once you've got your papers sorted, you give us some insight into how easy or hard or near impossible it has been reaching your decision only marginally quicker than the OJ jury did.
That was horrific. We ended up doing the black hole rock paper scissors.
Right.
So
Spock Lizard. Yes.
Yeah.
Fine.
Yeah. No, that was. Yeah, that was.
And that would be probably just as fitting in some ways because they were all brilliant. Yeah.
It was. I think. Safe to say that was very, very hard, which is why
you were long. Yeah.
We took a very long time, so I'm really, really sorry.
We didn't notice because Catherine was great.
Sorry about that.
Yeah.
And just when we thought we'd chosen, it turns out our choices were not as predicted, and we had to go back.
Yeah.
All right.
It was we deliberated long and hard.
But you did realise it was only these 10 you could choose from, didn't you?
I thought it was all Famelab since the beginning. Okay. So what was the question?
The question was, I can see why they took so long reaching a decision, can't you? Right. Yeah. The question was, maybe you could tell us a little bit about how difficult it was or whatever, and some general thoughts on the masses?
Well, it's, everyone was brilliant. That was the thing. We went and we sat down and we looked at each other askance and, well, how on earth do we sort out one being better than the other? Because it's comparing apples and oranges. It was almost, it almost got into proper fisticuffs.
Apples are better.
Apples are better, yeah, exactly, yeah. But we have a decision.
Okay, so let's start with your decision on the two joint runners-up. Can we get to the stage ready to present them with their prize? The head of communications at EDF Energy Nuclear Generation, and a man who's still 2 years on after I first pointed this out, tweets as 50-something but not quite grown up, Bob Fenton. Or maybe not. Oh no, he's here. Right, you see, it's not just this lot now to milk the applause, you see, you kept that going, fine. Right,
okay, so our first runner-up.
Of the two who are equal.
Of the two who are equal, yes.
Yes.
So in no particular order.
Exactly, but it's hard, it's one of those linear things, you can't speak in parallel, we understand.
Yes. Bert.
That is such good news for his shelving, yes?
And our next, in no particular order, Jace.
Brilliant, well done. Brilliant. So now we find out whether there is concordance with the audience or divergence from the audience. This is not the X Factor or even the Great British Bake Off. We won't drag this out. Who won Famelab UK 2016 and why? Preferably not in that order.
The winner is Kyle.
Hey! Now, that was the bit, actually, where we now call up Louise to officially present him with this, but kind of Bob cunningly did it. But, Louise, we'll get you up on stage anyway. So, one more time, Louise Emerson, CEO. So, come here. A few small words.
Yes.
Was this in your mind? When you entered Famelab all those months ago, the thought of standing here with the non-poison champagne and the check and everything else?
Yeah, I have to check those bottles. No, not at all. I just wanted to have fun, really, and to talk about maths to more people. So yeah, I get to talk about maths in the day and in the evening now, so it's fantastic.
And would it be foolish of me to speculate that £750 for science communication activities might go on the next EP, getting a bigger release, an LP of maths-related songs, perhaps?
I would love to. I would love to do that. That would be fantastic. I don't know what I'm going to do, but I'm absolutely honoured that people enjoyed. Who knew the intersection of folk and maths would go down so well?
It's the Venn that's the fun that we like as well. And obviously, finally, presumably you want to say something to these losers who you'll never see again.
No, these guys have been amazing, had so much fun. And Helen, who's arranged all of this stuff for Famelab, I think Helen should have a round of applause as well.
We'll thank her about a good idea as well. Okay, brilliant. That, I'm afraid, is where this particular approved science communication activity has to go to an end, and we're going to give another thanks to Helen as well. But before the law of the pub gets too strong, can we please again thank Louise Emerson, CEO, Cheltenham Festivals, and also everyone at Cheltenham Festivals. The British Council, who of course have been with us since almost the very beginning. Bob Fenton and everyone at EDF Energy, our associate title sponsor. Well, everyone apart from that weird guy in security you've got. I want to talk about him later. That's made up. Hazeltide Lake, our partners, all the local partners, including numerous universities and science museums who put on all the regional heats and put a lot of work in. All the trainers, the cerebral Cerberus that did our judging, namely Dallas Campbell, Mary Ryan, Ben Garrett. Poison penner Dr. Catherine Harkup, wherever she may have gone. The aforementioned fabulous Famelab manager, Helen Kirkman. Chris Tarrant, who's led all the backstage stuff tonight. Everybody who's been watching this live streamed all over the world, thank you for bearing with us and sorry if there's any dodgy signals. And of course, all of you in the audience for coming along. Thank yourselves. And finally, all 10 of our Famelab finalists. I think without exaggeration, we can say that today has been a vital, important and unforgettable day in the history of science. It was on April the 25th, 1953, that Watson and Crick first had their DNA double helix published in Nature. But it's also been not a bad day for Famelab. And our new champion, Kyle. Good night and go home.